Hello and welcome to my histology series. The video in focus will be looking at the respiratory system histology. And the first segment, we're going to have a short discussion on what we expect to find in the respiratory system. Then the last segment, I'm going to show slides and a bit of a description of the slides that are going to be of various structures within the respiratory system. So to begin with, we'll start with the nasal cavity, where you have the nasal vestibule, which is simply the dilated spaces of the nasal cavities, which are just inside the nasal nostrils. And those are going to be lined by vibrissae hairs. And in terms of epithelium, there's going to be a stratified squamous epithelium. Then there's a respiratory portion and an olfactory portion within your nasal cavity. The respiratory portion, I would describe it with the rest of the respiratory system. Then the nasal portion will be lined by a specialized epithelium, which is going to be olfactory epithelium. Then if you look at the vestibules, you expect to find sweat glands and sebaceous glands. The sweat glands and the sebaceous glands, in particular, if you look at the sebaceous glands, their secretions will actually be important in trapping particulate material that actually gets into the nasal cavity, right? Then the olfactory region, it's going to be located on part of the dome of each nasal cavity, and you find a specialized olfactory epithelium, which typically has 3 cells. And in the living, that mucosa of the olfactory epithelium will have a characteristic color which is almost yellowish brown due to the pigment that you find in the supporting cells and the olfactory glands, as well as the olfactory epithelium, the one that I'm going to describe shortly. Right. Then the surface area, or the total surface area within this olfactory mucosa is going to be about 10 square meters, as opposed to the 75 square meters that we find in totality for alveoli. Right. So the olfactory epithelium, like I said, is going to have 3 types of cells. So you find the bipolar neurons, which are your olfactory receptor cells. Remember from your general histology, when you did nerve histology, or you trace back in memory lane or back to physiology, where you say that you have 4 types of cells in the nervous system in terms of the neurons. One of them was the bipolar neurons, which you said you'd expect to find in spatial senses. The bipolar neurons would have 1 axon and 1 dendrite, right? So one of the spatial senses would be the olfactory epithelium for olfaction, and you find those bipolar neurons. Then you have the supporting or sustentacular cells, which support the bipolar neurons, and these cells are going to be columnar cells. Right? And they actually provide mechanical support and metabolic support to the bipolar neurons. Right? Then you have the basal cells, which are basically for renewal. So they act as stem cells for the olfactory cells, as well as the sustentacular cells, which are your supporting cells. Right? Then within this olfactory epithelium, if you look at the lamina propria, remember the lamina propria will be rich in glands known as olfactory glands, or your Bowman's glands, right? And this lamina propria will actually be directly contiguous with the periosteum of the underlying bone, because there is no submucosa or muscularis layer within this region, right? And if you look at the supporting cells, remember earlier when I said these supporting cells will actually have a pigment within them, just like the olfactory glands, which will give this olfactory mucosa a yellowish brown appearance in the living, right? Then they have numerous alveoli and on the apical end, as well as microvilli, right? Then if you look at the basal cells will be small and rounded. When I show you the picture later on, you're going to see that the cells that are closest to the surface in this olfactory epithelium, they're actually going to be the supporting cells or your sustentacular cells, and the basal cells are more basal, right? And those are actually going to be very small, and they're going to be rounded cells, and they act as stem cells, like we said, right? Then your olfactory glands, or the Bowman's glands, they're going to be tubuloalveolar serous glands, and they're of the branched type. meaning they are compound tubular alveolar glands, right? And the secretions that they actually secrete, they're going to be proteinaceous, meaning it's a serous gland, right? Then within these olfactory glands, you find lipofuscin, which is one of the pigments that you find from general histology or cellular inclusions, right? And this lipofuscin, which is in the gland, as well as the lipofuscin granules in your supporting cells, they're the ones that give the mucosa that yellowish brown coloration that I've been talking about, right? Then before we talk about the respiratory epithelium itself, you also have paranasal sinuses, which are going to be extensions of this respiratory system. And we describe those, or we name those in accordance to the bone that you find them. For example, you have the maxillary sinuses, you have the frontal sinuses, which are going to be asymmetrical. Then you have the sphenoidal sinuses in the sphenoid. Then you have ethmoidal sinuses, which you divide into anterior, medial, and posterior, right? These paranasal sinuses are going to be lined by respiratory epithelium, right? And they're subjected to acute infections by viruses leading to sinusitis, right? Then you also have the pharynx. The pharynx will also be part of the conducting portion, and you're also going to expect to find respiratory epithelium there, right? Then you have the larynx. But before we talk about the larynx, let's now talk about the respiratory epithelium, which we said it lines the respiratory portion of the nasal cavity all the way down to the bronchi. Right? So this respiratory epithelium, just like the olfactory epithelium, it's going to be a pseudostratified ciliated columnar epithelium. Right? And this pseudostratified ciliated columnar epithelium is going to be characterized by the presence of 5 cells. And these cells are actually going to be different in accordance to the region where you are. So you have ciliated columnar cells, which are the most abundant cells in respiratory epithelium. And on the apical end of each, you find about 250 to 300 cilia on the apical surface. Remember the apical surface modifications in epithelium from general histology. Then you also have the goblet cells, which secrete mucus. Then you have the basal cells, which act as stem cells. Then you have the Bright cells, which we're going to describe as having irregular microvilli. And on the basal end, those cells are going to be in contact with axons of nerves. Hence, those are going to be chemosensory cells, right? And they resemble the gustatory cells for taste. Those Bright cells make up 3% of the cells that you find in the respiratory epithelium. Then there are the small granule cells of Kaushitsky, which are part of the diffuse neuroendocrine system, and they're going to secrete things like catecholamines and so forth. Those small granule cells are also going to make up 30% of the total cells that we find in our respiratory epithelium. Right? So that's the epithelium that you find on the respiratory portion of the nasal cavity. That's the same epithelium that you expect to find in the pharynx. Then we get to the larynx. The larynx is going to have respiratory epithelium, except over the vocal cords, where the epithelium becomes stratified squamous. I'm going to show you a picture of the larynx towards the end. Right. Then the trachea also have respiratory epithelium, but this time around with 20 C-shaped rings of cartilage that is going to be hyaline cartilage. Right. But if you look at the trachea in terms of its layers, it's going to have a mucosa, which is that ciliated pseudostratified columnar epithelium. And then the lamina propria of the trachea will be rich in a fibroelastic material. So it's a fibroelastic lamina propria. Then the submucosa will be slightly dense connective tissue as compared to the lamina propria, where the connective tissue will be smooth, or will be loose rather. Then below the submucosa, we find the cartilaginous layer, the one that is composed of those C-shaped hyaline cartilages. Then the adventitia will actually be composed of connective tissue, which is going to bend or blend with adjacent structures within the thorax, right? Then for the epithelium, remember we talk about metaplasia. Metaplasia is change from one adult form of epithelium to another, and it's actually reversible. The metaplasia that is more common in the respiratory system, it's going to be a squamous metaplasia, which is why we afford to talk of squamous carcinomas in the respiratory system. It's from those squamous cells that as a result of the metaplasia, right? And it's due to subjecting our respiratory epithelium to stress. And if we subject our respiratory epithelium to stress, it's going to change in such a way to become an epithelium which is adapted to the ensuing stress. Right. Then for the trachea, remember the cells that are going to be there, say they're mostly your ciliated columnar cells, and those are going to be the most abundant cells. The purpose of the cilia is to provide a sweeping motion for the mucus coat, so that you actually move it all the way to the nasal cavity. So it forms part of the mucociliary escalator. Right. Then the mucus cells producing cells in the trachea will be still the goblet cells, which we say they secrete mucus in the respiratory epithelium. Then I talked about the brush cells. They're also going to be there in the trachea. And on the basal end of these brush cells, remember I said there is synaptic contact with afferent nerve endings. Then you also have the small granule cells. I talked about those as being part of the diffuse neuroendocrine system. They produce things like serotonin, catecholamines, and so forth. You get basal cells. The basal cells act as stem cells. Right. Then you have a basement membrane. Then you have a lamina propria, which is said is fibroelastic. The basement membrane is actually very, very thick for the trachea as compared to other regions. Then we talked about the submucosa having a more dense connective tissue. Then you also have lymphatic tissue that is also in this lamina propria, which forms part of the bout, your bronchus-associated lymphatic tissue. Right. Then we talked about the tracheal cartilage, 15 to 20 C-shaped rings of cartilage, which other books describe as being U-shaped. Then talked about the adventitia. Right. Moving on, we get to the bronchi. The bronchi are also going to be lined by respiratory epithelium, but notice within the bronchi, the epithelium still going to be respiratory. Then when you get to the bronchioles, the epithelium starts to transition from a respiratory to a simple epithelium, which I'm going to describe. The cartilage also transitions from the trachea, where we said it is almost regularly arranged into those C-shaped rings of cartilage. In the bronchi, they're going to be irregular plates of cartilage, right? And remember, the bronchi will be divided into, there's the right and the left primary bronchi, which divide into lobar bronchi. Then you get to segmental bronchi, right? Your tertiary bronchi, right? Which are going to be for the bronchopulmonary segments, right? For the bronchi, the mucosa is going to be still the respiratory epithelium, which is also the stratified epithelium. Then you also expect to find the lamina propria, which is going to be the same as the trachea. It's going to be a fibroelastic lamina propria. Then you also expect a muscularis, which is going to be loosely arranged. The muscularis, as you can see on the board, it's going to be spiral shaped smooth muscle. Right? Then you have the submucosa, which is relatively loose connective tissue as compared to the trachea. And you're going to have glands as well as adipose tissue going to be present within the submucosa, particularly of the larger bronchi, right? Then there's also a cartilage layer. The cartilage layer, like I said, is no longer going to be continuous. They're going to be irregular plates of cartilage. Then you also have an adventitia, which is moderately dense connective tissue and is also continuous with the adjacent structures to include the pulmonary vessels. Right. Then from the bronchi, you move to the bronchioles. The bronchioles, they have simple ciliated cuboidal, which transitions or which has transitioned from the respiratory epithelium, which at some point you'd see as simple columnar, and ultimately the smaller bronchioles will have simple cuboidal epithelium. right? You're going to find Clutter cells, and which these Clutter cells can also be called exocrine bronchiolar cells. There is no cartilage, specifically in the smaller bronchioles, and there is prominent smooth muscle, right? And for the larger bronchioles, there are going to be branches that are going to be emerging from the tertiary bronchi, which we call the broncho, which are actually going to be the segmental bronchi for the bronchopulmonary segments. Right. Then for bronchioles, we don't expect to find cartilage. We also don't expect to find goblet cells, but those goblet cells in the largest bronchioles, you still have them, but in the smaller bronchioles, they disappear. Right. Then the Clutter cells, they're going to be non-ciliated cells, and they have a characteristic round shape, those Clutter cells. They are simply non-ciliated cells, and they secrete a serous fluid. Right? And those Clutter cells are found in the bronchioles. Serous secreting cells have characteristics of protein secreting or protein synthesizing cells. So expect to find rough endoplasmic reticulum, expect to find the Golgi apparatus, you expect to find a nucleus, which is euchromatic, which typifies an actively synthesizing cell, right? Then if you look at the terminal bronchioles, we say the epithelium becomes simple ciliated cuboidal with Clutter cells that are not going to be ciliated. There is scattered alveoli. There is still no cartilage. There is incomplete circular smooth muscle as compared to the larger bronchioles, which have prominent smooth muscle. Then the respiratory bronchioles have simple ciliated cuboidal with Clutter cells. Then you have alveoli present, no cartilage, and a few muscle fibers. Then you have the alveoli, which are the sites of gaseous exchange. But before you get to the alveoli, there are the alveolar ducts. Then you also have the alveolar sacs. Right? The alveolar ducts are elongated airways that have almost no walls, and they're only made up of those alveoli. And the alveolar sacs are simply spaces that are surrounded by clusters of alveoli. Right? The types of cells that you find in the alveoli, you have two types of pneumocytes, type 1 and type 2. The type 1 pneumocytes make up 40% of the total pneumocytes that you find in the respiratory alveoli, and they line 95% of the surface, right? Those type 1 pneumocytes cannot divide. So they have lost the ability to undergo mitosis, and those cells are actually going to be simple squamous cells, the ones that allow for gaseous exchange, right? Then the type 2 pneumocytes, or the type 2 alveolar cells, are also going to be septal cells, and they're secretory. Those cells are cuboidal, and they have a rounded nuclei. Those type 2 pneumocytes secrete surfactant, which you know from your physiology, surfactant lowers surface tension, right? And the surfactant will be stored in what are known as lamellar bodies within those type 2 pneumocytes. So you find lamellar bodies storing the surfactant within granules, right? And there's an interalveolar septum which exists between alveoli. The interalveolar septum will have elastic fibers. It will also have reticular fibers and alveolar macrophages. Some of the alveolar macrophages will actually be found within the alveolar, and they'll actually remove the dust that you find within the alveolar, and those are hence called dust cells. Then you also remember that the alveoli are actually going to communicate via alveolar pores, which are known as the pores of Kohn, right? Then the surfactant will have things like phospholipids, hydrophobic proteins, and so forth. But the proteins of importance, there is surfactant protein A, which is SPA, there is B, there is C, and there is D. But most importantly, D is involved in host defense mechanisms in the innate immunity. So they actually fight gram-negative bacteria, right? Then together with surfactant protein A, they actually work in the allergic response to inhaled antigens that you must have mentioned in your physiology. Right. Then the alveolar macrophages are a bit unusual in the sense that they're not only in the connective tissue, like I said, you can also find them within the alveoli. So they're also going to bulge within the lumen of the respiratory system, which is the same thing that you expect to find with the clutter cells. They also bulge into the lumen of the bronchioles, right? And clinically, you can talk about infantile respiratory distress syndrome, also known as hyaline membrane disease. So you have a surfactant problem and you have surface, an increase in surface tension. You can talk of alveolar collapse, that's atelectasis. You can talk of cystic fibrosis, which is an autosomal recessive disorder with a mutation on chromosome 7, which affects the CFTR channel, which is the chloride channel, and it's going to present with a sticky mucus, and it also affects the sweat and the saliva. Right? You talk about emphysema. Emphysema is a condition where you have permanent enlargement of the air spaces distal to the terminal bronchioles. Right. Then there's cancer of the respiratory system. There is small cell cancer. There is large cell carcinoma. Yeah. There is adenocarcinoma, which is more in females. Then there's the squamous cell carcinoma, which is typical or mostly found in smokers. Right. Those are all forms of bronchocarcinoma. Then you also talk of bronchiectasis, which is overdilation of your bronchioles, right? Then the next segment will then look at the pictures that will show the respiratory structures from a histological perspective. So these here are slides from the nasal cavity. So this is a low power where we can appreciate that this is the mucosa of the olfactory epithelium. Then this here, this is going to be part of the ethmoid bone. Right. If we look at this mucosa, the mucosa will have an epithelium, just this one. That is going to be olfactory epithelium. Right. Then you also expect to find glands that I'll show you at higher magnification. This here from this other end, this is respiratory epithelium. This is olfactory epithelium. And remember, I said it's a mucoperiosteum. This mucosa rests directly on bone, right? And these are just this is just a defect from the slide where you see a slight separation between the mucosa and your periosteum, right? Then if you come to a higher magnification, this is going to be the epithelium. So these cells that have nuclei that are dense and closer to the surface of the mucosa, those are going to be the supporting cells, like we said earlier on, the sustentacular cells. Then these basally located cells here with rounded nuclei, those are going to be the basal cells for regeneration of the bipolar neurons as well as the supporting cells. Then these ones, these are going to be your bipolar neurons, right? The olfactory cells. Right? So those are your olfactory cells. And if you look at this, right, this here is a nerve. Right? Then this here, that's a Bowman's gland. Right? That's another Bowman's gland. That's another Bowman's gland. That's the lamina propria. That's another Bowman's gland. Right? Then if you come here, that's another supporting cell. Right? Then from here, that's a vein. That's a capillary. Remember, capillaries are one cell thick. They only have an endothelium supported by pericytes. So that's a capillary there. Right. Then, so basically this is the mucosa of the olfactory epithelium rather. Those are the three types of cells in the epithelium. This is the lamina propria with the Bowman's glands. Right? Then you also find blood vessels, you find nerves. The blood vessels are for you to actually warm the air that you're actually taking in. Right? Then there's a slide of the larynx. If you look at the larynx, these are going to be the ventricles. Right? This is low magnification. If you come to the bottom slides here, this is the epithelium. Remember I said the larynx will have respiratory epithelium, but in places where you find the vocal cords, you have a stratified squamous epithelium. Right? So this is a stratified squamous epithelium on this side. This is a stratified squamous epithelium on this side. That's a gland. Remember we mentioned there are glands in the lamina propria of the larynx, as well as some parts of the respiratory system. So that's a gland. That's stratified squamous. That's stratified squamous epithelium. You come this side. This is stratified squamous epithelium. This now typifies respiratory epithelium on this side. This epithelium is now pseudostratified ciliated columnar epithelium. That's a gland in the lamina propria. That's another gland over there. Those are glands. That's a gland again. This is pseudostratified ciliated, different from the stratified squamous epithelium on the side. Right. Then we get to the trachea. This is the trachea. This is the pseudostratified ciliated columnar epithelium, the respiratory epithelium. Right. So that's your epithelium. Right. And if you look at the lightly staining regions in the epithelium there, that's where you find goblet cells. Remember, goblet cells who have a basally located nucleus, and on the apical end, you expect to find mucins. Mucins stain lightly under the light microscope, and when preparing slides, they're actually squeezed out of your epithelium, right? Then this is lamina propria, right? Then remember I described a thick basement membrane, right? This one in your trachea, right? Then this is smooth muscle. Right? That's going to be smooth muscle there. Then if you look at this, that, and that, that's going to be your glands. Right? Then this here, take a moment to pause. Remember, we said the tracheal layers, we talked about the tracheal layers, right? This is part of trachealis, the trachealis muscle. These are all glands, these ones in the lamina propria, and some of them extend into the submucosa. Above this muscularis layer, that will be the submucosa, right? Then these are also glands, right? Then looking at it from a higher magnification, this is the epithelium, that's a goblet cell. Those would be the columnar cells, but that's a goblet cell there with a basally located nucleus. Right? The nucleus is basally located there. If you can look at the cursor, right? This is epithelium, right? You can see the cilia on the apical ends there, clearly visible on the middle insert. Right? You can still see the thick basement membrane there. Right? From this side, we can appreciate the submucosa. We can appreciate the presence of this structure here, which is a lymphatic nodule. Right? Then from this side here, we can appreciate this here, which is cartilage. If you remember how cartilage looks like under the light microscope, this is cartilage, tracheal cartilage, right? Then if you come this side, this here is a vein, right? That's a vein right there. So basically this is how the trachea looks like. Then If you go to the bronchioles, this is a bronchiole, right? And remember, we said this is the epithelium. That's your bronchiolar epithelium, right? Then this is going to be smooth muscle surrounding it. Right? That's smooth muscle. Then what else can we see here? Right there, those are blood vessels. This is an alveolar duct. Right? These are alveolar sacs. Remember I said alveolar sacs. There will be concretions of alveoli. That's an alveolar sac, that's an alveolar sac. These are the alveolar ducts. Right? Then these are now respiratory bronchioles, these ones from the bottom slide. Those are respiratory bronchioles. These are nodules of lymphocytes, which we say they form part of your bout. That's lymphoid tissue, right? Then all these are alveolar sacs, right? Then this here is the lymphoid tissue that I just mentioned, right? Then this is more of the respiratory bronchioles, but we can appreciate that this year, forget what's on the left, look at this on the right. This year, this is going to be an alveolus, right? And this here is those are clutter cells in the bronchiolar epithelium, but this is an alveolus, right? Then what else can we see? Those are the simple squamous cells lining the alveolus. Then maybe if we use the bottom one, right? Then that's a capillary. Right? That's a capillary right over there. That's an alveolus, that's an alveolus, that's an alveolus, right? That's a septal cell between alveoli, right? Where we said you also expect to find macrophages, right? So that's just about it. Thank you for watching.